Okay, Patrick, you have a question. Hello, everybody.
Hey, Professor, can you hear me?
Hello.
So I followed the instructions very clearly. I
Okay,
don't know if
so
I'm
let's make a deal. I will answer all the questions on exam soft at the end of lecture, okay?
okay, no problem.
I can cover the lecture material, and then I promise I will answer all the questions you have, okay?
Sure, thank
Is that a
you.
deal?
Deal.
Good. Oh, you cannot hear? Now can
I
you
hear
hear
you,
me?
Professor.
You can hear me now.
Yeah, we can.
Okay, great. Okay, so as I said to Patrick, I will be glad to answer all your questions on exam soft, but at the end of the lecture. Okay, so exam soft. It's not so scary. You know, I had to be trained on this new platform, but I think now I know what has to be done, and I'm not so scared anymore. So everybody, remember that the day after tomorrow, it's going to be September 11, from 4 to 5:15, we'll have our mock exam. And I will also, if I have time, I will also cover some lecture material. It shouldn't take us too long because this is just a mock exam so that you become familiarized with exam soft. So everybody has to be at the 68th Street campus, which is where I am right now. The exam will be on the first floor, Assembly Hall, so you don't have to depend on the elevators. Everything will be good. And then there's in-person attendance and there's no grades because it's just a mock exam so that you'll be trained. So by now, all of you should have received an email to download exam soft. So you have to be invited because you don't pay, it's free for the students, but the school pays for it. So you have to be invited to download exam soft. If you can't log in to ExamSoft, make sure that you use this email because some students cannot see the invitation. That's because you're looking at in your Gmail or Yahoo or whatever. So Hunter sends out information to your My Hunter CUNY EDU email. So make sure that you look there. The mock exam is already uploaded and it's ready. So, you know, it's there. So you can try to take it and access it more than once. So that when the real exam comes on September 25, all ready to download the exam and to take it without any problem. So for the real exam, you'll have a password that you will only know when I release it. But for the mock exam, you can have a password because it doesn't count and it's just, as I said, a training mechanism. So this is the password that you will use to have access to the mock exam. Please make sure that ExamSoft is installed on your smart device when you come on Thursday. Don't wait until you are in the auditorium to download ExamSoft. Come with it already downloaded. If you have problems, they have an amazing tech assistant service. So you can either call them by phone or you can visit the ExamSoft support site. And on the right corner, you'll see a chat box where you can ask all kinds of questions. So this is a wonderful service that will help you with the download of the ExamSoft, and also if you have problems looking at or finding the mock exam. So right now, I don't want to say anything else about the ExamSoft, and as I said, I will answer all the questions at the end. Okay, so today we fit in the previous lectures we covered chemistry. Now we're shifting to cells, and the study of cells and all their properties. So you can see here drawings of all different types of cells. Now, the point that I want to make is all these cells have exactly the same genes, the same genome. Genome is a collection of genes that make up the human genome. So, why is it that if they all have the same genes, how come they have so many different morphologies and so many different properties? Because humans have around 25,000 genes. But not all 25,000 genes are going to be expressed in the cells, in all cells of different types, and not at the same time. Some genes can be expressed when cells are under normal conditions, and other genes will be expressed under stress conditions. So although the cells share the expression of many genes, but some are specific for neurons. This is a beautiful cell, it's a neuron. Here's smooth muscle. Here's the epithelial cell that makes up the walls of the small intestine, the red blood cells, and the sperm. So they all have the same genome, but what we call transcriptome, which are the genes that are actively being expressed, they differ from cell type to cell type. And why am I showing here this tree with so many leaves? So this tree is about 60 to 70 years old, and it has about 175 to 225,000 leaves. And each leaf has to receive the proper nutrients, proper amount of water, proper oxygen and CO2. So everything is controlled in each of these leaves. Now, in our body, we have 75 to 100 trillion cells, not 1,000, trillion cells. And all the cells have to be maintained under homeostatic conditions. So each cell has its own regulatory mechanism to maintain homeostasis. And remember that different types of cells are organized into organs, and the organs into systems that we are going to study some of them. So all this is really interesting and fascinating, and there's a lot to learn. Although there's lots of research throughout century, but there's still a lot to be learned. And now we have the ability to, for example, you take out a cell from the skin and de-differentiate it and into a pluripotent cell. And that pluripotent cell then can be differentiated, for example, into a neuron. This is the type of research that I'm doing now in my lab. So we have cells that were taken from a normal individual from the skin, they're called fibroblasts, and then they were de-differentiated into pluripotent. Pluripotent, that means that they can be differentiated, they can give origin to different types of cells. And we are studying these cells that are converted into neurons. And that's our model for Alzheimer's disease. So you see here, the beauty of this is that there's so many different cells. There's 200 different types of cells in our body. They all have to be maintained under homeostatic conditions. And this depends, remember, on the nervous system and the endocrine system. Okay, so what's a cell? A cell is an amazing structure that contains what we call organelles. And each organelle has its own function. And its function depends on its contribution to homeostasis. So, for example, we have mitochondria that produce ATP. We have peroxisomes that get rid of hydrogen peroxide. Then we have lysosomes that are the stomach of the cell. They digest the nutrients and so on. And outside of this cell, inside is called the cytoplasm, and outside we have the plasma membrane. And that's what we will be discussing today, the plasma membrane of the cell. And then in future directions, in the future lectures, we will discuss the different organelles. But remember that this all together, you could say that this is almost like an organism, because it is not an organism. An organism is made up of thousands and trillions of cells. But here, each cell has its components that contribute to the maintenance of homeostasis. And now let's see what is the function of the plasma membrane. So the plasma membrane is a membrane, as its name indicates. And what is it made up of? It's made up of phospholipids. So you can see, remember when we discussed phospholipids, it's an amphipathic, phospholipid is a lipid, and it's an amphipathic molecule. Why? Because it has a charged moiety here represented by these blue spheres, and the tails are uncharged, or let's say polar and unpolar. And the plasma membrane is made up of a phospholipid bilayer. Bilayer means two layers of phospholipids. And you can see here that the membrane is not static, it's not a rigid structure, it fluctuates. And embedded in this structure made up of phospholipid bilayer are these proteins that are shown here in red. And these proteins have all kinds of shapes and functions. So look at this protein. It has a spaghetti-like structure, and within the area, this is the hydrophobic area of the plasma membrane. You see that it has this hydrophobic helix that keeps it or anchors it to the membrane. Others have this globular structure. So there are all kinds of proteins. We have 25,000 genes, but in our cells, there's around 100,000 different types of proteins. Now, an interesting aspect that is shown here is, let's say, this part of the membrane or side of the membrane is facing the extracellular space, so outside the cell. So here, that's the extracellular space, and inside is the intracellular. So here, this side of the membrane is facing the extracellular environment. And you can see that it has these hexagons that represent carbohydrates. So that means that the proteins that are embedded in the plasma membrane on one side, they can be decorated by carbohydrates. But the side that faces the intracellular space does not have carbohydrates attached to the proteins. So here you already see that there is an asymmetric structure of the plasma membrane. The extracellular domain is different from the intracellular domain, and that also depends on the composition of the phospholipids. So here you see what else is in the membrane. So phospholipids are the major components of the membrane. They make up 75% of the plasma membrane. We also have glycolipids. So not only are proteins decorated by carbohydrates, but also lipids are decorated by carbohydrates. You see here, this blue structure represents a carbohydrate. So here you have the tails, and instead of having a charged group, a polar group, now you have carbohydrates. So this lipid that is decorated by a carbohydrate is known as glycolipid. The same as if a protein is decorated by a carbohydrate, you say it's a glycoprotein. So the composition of the phospholipids on the extracellular domain is different from the phospholipids that are present in the intracellular domain. So you see here the phospholipid polar heads are represented in all different colors. Why? Because they're chemically different. So the composition of the extracellular layer is different from the composition of the intracellular layer. This is in terms of phospholipids. Okay, so 5% are glycolipids. Will we have to know the parts of the cell membrane and their functions? Yes. Is membrane lipids and plasma membrane the same? No. Membrane lipids are components of the plasma membrane. No, you have to know phospholipids, glycolipids, and cholesterol. Yes, you have to know what that is, but not the name of each of them.
Professor,
So why is cholesterol? Yes?
are you saying that 75 of the plasma membrane is made up of 75% phospholipids?
75% of the membrane is made up of phospholipids.
Okay.
That's what is represented here and is represented here. The blue heads are the polar part of the phospholipid, and then facing the inside are the tails. So 75% of the membrane is made up of phospholipids. Okay, I'm not going to answer exam soft questions. Okay, if you can wait until the end, then I will answer all of them. So that means that the plasma membrane is asymmetrical. It's asymmetrical because the phospholipid components of the outer layer are different from the phospholipid component from the inner layer. Also, cholesterol gives the membrane rigidity. And cholesterol makes up 20% of the plasma membrane. So we always think that cholesterol is a bad lipid, it's a bad molecule, but it's not. It has very useful properties. One of them is to maintain the plasma membrane rigid. Okay, so membrane proteins we were talking about. You see here that some of them go through the phospholipid bilayers. So you see here, these ellipses represent the polar end of the phospholipid. And here you have the tail, so there's two layers, and the tailors are arranged. So please, can you mute yourself?
Please mute yourself.
Okay. So here you have these proteins that span the two phospholipid bilayers, so they go from the outside to the inside. These are known as transmembrane proteins, and they're very useful because they serve as channels. Okay, but there are other proteins that are only attached either to the outer layer or to the inner layer. And how are they attached? They're attached by phospholipid tails and all kinds of different types of attachment to maintain these proteins in place. So a transmembrane protein is a protein that crosses the two phospholipid bilayers. So it can be used, for example, as a channel. So let's say a sodium ion can go through this channel or go from the outside to the inside, because this channel is a transmembrane protein that crosses the phospholipid bilayer. Okay, so what are some of the functions of the proteins that are associated with the plasma membrane? So one function, as I just explained, they are channels. Well, let's say they are pores, so that polar molecules, for example, ions that are charged, like sodium is a cation, potassium is a cation, so they would be repelled by the polar heads of the phospholipid. So how can they get into the cell? They can get into the cell through, yes, aquaporins or transmembrane proteins. So they get in through these channels that can span cross the two phospholipid bilayer. Other proteins are transporters. So the transporters, think about glucose. Glucose is a polar molecule and it cannot get into the cell freely. So how does it get into the cell? One way is through these transporters. So glucose binds to one of these proteins. This protein, which is a transporter, changes shape and allows glucose to go in. Other proteins are receptors. What does that mean? That they are chemicals that cannot cross the phospholipid bilayer, but how do they confer? How do they send out the signal? So they bind to the receptor, and then the receptor changes shape, and then it sends a message to the inside of the cell. That means this ligand will not go into the cell like glucose. Glucose gets into the cell, but there are chemicals that don't get into the cell. What happens is they bind to a receptor, and then the change in shape of that receptor transmits the message into the inside of the cell. Or transporters receptor mediated? Well, it depends. There are all kinds of ways to regulate these transporters. And you will learn, for example, insulin. Insulin will regulate the number of transporters that are at the plasma membrane for glucose. So and I will explain this shortly. Other membranes, other proteins are cell identity markers. So what do I mean here? Remember the ABO blood group? So this ABO blood group was dependent on the carbohydrate that's attached to the protein. So that's what a cell identity marker is considered. So it's a protein that's attached like a glycoprotein that's attached to the carbohydrate. And this type of carbohydrate identifies what type of cell this is. Other proteins are involved in cell adhesion. So they allow cells to be attached to each other. So they play a role on cell adhesion. Adhesion means attached. Another are enzymes. Remember, enzymes are proteins that make chemical reactions more efficient. So you have the substrates that bind to the enzyme, to the active site of the enzyme. They come together to form a more complex molecule. And the enzymes are not used up. They're catalysts, remember, they're not used up in the chemical reaction. So here are some examples of the functions of proteins that are attached to the plasma membrane. So as I said before, the plasma membrane is not a rigid structure. So you see here, these look like ping pong balls, these spheres. These are the polar heads of the phospholipid. So this would be a phospholipid with its polar head and the two tails. Remember that the backbone is glycerol. So you have two fatty acids, right? And you have a polar group. But you see that this flows around, and the proteins that are transmembrane also move around within this phospholipid bilayer. And actually, there are chemical reactions that can occur within this hydrophobic space that is generated by the alignment of the phospholipids. So you see that the outer side of the membrane is charged, is polar, and so is the inner layer. But the middle is hydrophobic in its not charged. Okay. Now, another interesting aspect that I already sort of explained is that the outer layer of the plasma membrane is different from the inner layer. So the outer layer contains proteins and lipids that are glycosylated and forms what we call a glycocalyx. So you see here an electron micrograph that shows the plasma membrane, and this sort of hair-like image corresponds to carbohydrates that are attached either to lipids or proteins in the outer layer of the membrane. Now, the composition of the glycocalyx is specific for cells. So some cancer cells have a different collection of carbohydrates attached to the outer layer. So the nature of the glycocalyx can indicate if a particular cell, if a particular group of cells is cancerous, depending on what are the types of carbohydrates that are attached to the outer leaflet of the plasma membrane. So that is known as the glycocalyx. Glyco, because it's glycosylated, it's carbohydrates, and calyx means the outer layer. Okay, so the plasma membrane. Why do we need a plasma membrane? Well, we need a plasma membrane to define the shape and the cell of each cell. But it's also a very, very important selective barrier. And we have not, in research and investigators, have not been able to mimic the properties of the plasma membrane. So it's selective because it allows molecules to cross it depending on their charge and also on their size, but not only on their size. The size would be a semi-permeable membrane. Semi-permeable membrane is easier to reproduce, to mimic, than a selectively permeable membrane because one depends on just the size of molecules that can go in, and the other one, the selectivity depends on the charge of the molecules. So what can go in without any barriers? So gases, carbon dioxide and oxygen can get in very easily. There's no barriers because they're small, they're uncharged, and this is very important because it's oxygen and carbon dioxide are critical for maintaining the cell, the life of the cell. And oxygen is very important to make ATP, but also some reactions require oxygen. And carbon dioxide has to be removed. It's sort of a toxin that is produced in the cells. So carbon dioxide and oxygen can cross the plasma membrane freely. There's no barrier. And so do hydrophobic molecules. Hydrophobic, that means that they are soluble in lipids and not soluble in water. They have the phobia of water. They don't like water. They're hydrophobic. So benzene, for example, is a molecule that is lipid soluble. So it can cross very easily the plasma membrane. What about small polar molecules, such as water and ethanol, alcohol, that we drink, that is that we can drink freely, some of us, not too much, but the ethanol goes freely in and out of the cell. And if you have too much ethanol, sorry?
58851.
So ethanol and water can freely cross the plasma membrane, although they're polar, but they're small enough. And the movement of water is helped by aquaporin. Now, what about large polar molecules? So large polar molecules such as glucose. Glucose is essential also for the cells to live, but its movement across the plasma membrane is highly regulated. So it cannot, because it's large and polar, it cannot cross the plasma membrane. And that's why it needs transporters. Remember that I mentioned the transporters for glucose. So glucose, to get into the cells, requires special proteins that are transmembrane proteins that are transporters. Okay, and then what about small molecules that are charged, molecules and ions? So here you have hydrogen, chloride, sodium, calcium, potassium. These are all charged, negatively or positively charged. Also, amino acids, these cannot get into the cell because they are charged. So they need channels. They need channels to get in and out of the cell. So these glucose needs transporters, and these small charged molecules need channels. So why large polar molecules don't go through the plasma membrane? Because you have the heads of the phospholipids that are polar, and they will repel these polar molecules. So the charge, because they are charged, they are going to be repelled. So again, why can't the charged molecules cross the plasma membrane? Because the heads of the phospholipids form a charged barrier that repels anything that's charged. Okay, so what needs to cross the plasma membrane? Nutrients, of course, such as vitamins, oxygen, all kinds of nutrients have to get into the cell. And waste has to get out, carbon dioxide, nitrogen, and so on has to get out. So there's this movement in and out of the cell to nourish the cell and to get rid of waste. Signaling molecules, some hormones that are hydrophobic get into the cell, such as estrogen, testosterone. They get into the cell, bind to their receptors, and they're called signaling because then they send a signal to the inside of the cell to generate some kind of a response. Fluids also are important. We have water that gets in and out of the cell, and then ions, sodium, potassium, calcium. All those are essential for cells to survive. So now we talked about how the plasma membrane is a barrier, but how is it that molecules go through cross this phospholipid bilayer? So there's two types of transport. We consider passive transport, that transport of molecules that only require kinetic energy. Remember what kinetic energy is. Kinetic energy is the energy of movement. So you have the plasma membrane, something hits it, if it can go through, it goes through, based on kinetic energy. And that's the passive transport. It is only dependent on kinetic energy, the energy of movement. One particular property of this passive transport is that the molecules and ions always move from a side of the membrane that has a high concentration to low concentration. So they sort of go down the hill. They go from high to low. So that is characteristic or important for passive transport. First of all, they depend on the energy of movement, kinetic energy, and whatever is crossing the plasma membrane always goes from the side that there's high concentration to a side that there's low concentration. Now, the active transport is different. The active transport, the molecules and ions, they go from low concentration to high concentration. So they're going uphill. So they need much more energy. And the kinetic energy is not sufficient. Now they need potential energy. And this energy, remember, comes from ATP, hydrolysis. So each phosphate group that is removed from ATP generates energy. So here you have the two general types of transport. Passive transport that depends on kinetic energy, and whatever is being transported goes from high concentration to low concentration. And that means the side of the plasma membrane where there's high concentration to the side of the plasma membrane where there's low concentration. Active transport, because you're going up the hill, you go from low concentration to high concentration, you need more energy. And that energy is provided by the hydrolysis of ATP, and it's known as potential energy. It's the energy that is stored in covalent bonds. Now, ATP is so important that if you inhibit the production of ATP by mitochondria, so mitochondria are the organelles inside the cell that produce most ATP. So if you inhibit this protein known as cytochrome C oxidase, that's part of one of the enzymes within the mitochondria that contributes to the synthesis of ATP, if you inhibit this enzyme with cyanide, that's lethal because there's no ATP to produce active transport. So if you don't have active transport, the cell cannot survive. So now we're going to discuss the different types of passive and active transport. So there's 4 different types of passive transport: filtration, simple diffusion, facilitated diffusion, and osmosis. Osmosis only refers to water. And remember that passive transport depends on kinetic energy, and whatever is moving goes from high concentration to low concentration across the plasma membrane. The active transport is the opposite, goes from low concentration, so goes up the hill, low concentration to high concentration, and depends on potential energy in the form of ATP. So there's 2 forms of active transport: active transport per se and vesicular transport. Okay, let's start first with the active transport. The first type of active transport that I want to discuss is filtration. And this is an amazing type of passive transport that occurs in the kidney. So we have at one time around 180 liters of fluid that needs in the kidneys that needs to be filtered. So what does filtration do? Filtration gets rid of waste products, and it's also used to absorb water and so on. So the entire plasma volume inside the blood vessels, this plasma has nothing to do with the plasma membrane. Now we're talking about the liquid that makes up the blood. So the plasma is about 3 liters. So we have 3 liters of fluid present in our body, and it's filtered through the kidneys 60 times per day. So the whole plasma that is part of the blood, that's in the blood vessels circulating in the body, is filtered, it's purified. So filtered, you can say it's a purification of the blood. So these 3 liters are filtered 60 times a day. So the blood goes on, flows around, and goes through the kidneys to be purified. So let's look at the unit of the kidney that is responsible for this filtration, for this purification. So here's the arterial blood, and here's a capsule that is the unit that is responsible for filtration in the kidney. And this cup is known as the glomerulus. It's known as the Bowman capsule. And you see here that the artery becomes thinner and thinner and thinner, and it forms these very, very thin blood vessels that are known as glomerulus. So they fit perfectly within this capsule. And this is where filtration is occurring. So what happens here? So the glomerulus has a wall, and inside it has fluid that is part of the urine. This is where the urine is formed. So you see here a higher magnification of this unit of the kidney that is responsible for filtration, for purifying our blood, the plasma component of our blood. So the walls of these capillaries is formed of endothelial cells, but the endothelial cells that are neighbors, they have pores in between. So that means that small molecules and ions can pass through these pores. So we say that these arterioles or blood vessels are what we call fenestrated. Fenestrated, fenestra, means window in Latin. So they have these windows that allow small molecules and ions to go through independent of their charge. So there's no barrier here. This is all dependent on size. So you have the heart that's pumping the blood and generates a blood pressure, and it's that blood pressure that pushes these molecules through these pores that make up the walls of the capillaries. So you can, this process contributes to the purification of the blood. So these waste products get secreted or released, that's a better word, get released into the urine. So you see here the urine has a yellowish, is represented in a yellowish color. So the urine contains these waste products that are filtered from the blood vessels that are porous here, they're fenestrated. Also, water can go in and out freely. So this glomerulus is the unit of the kidney that is responsible for filtration. And this filtration is dependent on hydrostatic pressure or the blood pressure that is generated by the pumping of the heart. So you pump the heart to generate this pressure that pushes the waste products through these pores that are made by endothelial cells. Okay, so this is filtration. Now, another type of passive transport is simple diffusion. So what is it's so simple because the molecules or ions are going to be hydrophobic. They can cross the phospholipid bilayer without any barrier, without any impediment. So they are moving around through the Brownian movement, and when they bump onto the membrane, they can get in or out. So this is known as simple diffusion. And this depends on the hydrophobicity of the ions or molecules. They have to be able to cross the phospholipid bilayer. They are hydrophobic, so there's no barrier. They depend only at kinetic energy, on kinetic energy. So what does simple diffusion depend on? On the concentration gradient. Remember, passive transport, the molecules go from high concentration to low concentration. So the more particles you have on the outside, the more particles will bump to the plasma membrane and go through. So it's dependent on concentration. It's dependent on temperature. Why? Because higher temperature increases the movement of these particles, so they have a better chance to hit the plasma membrane. It also depends on the size, because if they're larger, they will have a harder time to be pushed through the phospholipid bilayer. They depend on the lipid solubility or the hydrophobicity so that they can cross the plasma membrane. They depend on the membrane surface area. So if they are just trying to get through in a small area, or if there's a large area where they're bouncing around, so the larger area gives them a better chance to get in or out of the cell. And the membrane thickness. So these are 6 parameters that regulate or the simple diffusion. So examples of molecules that use simple diffusion to get in and out of the cell through the plasma membrane. So oxygen and carbon dioxide, they move through simple diffusion, but also some nutrients that are lipid soluble, such as vitamin D. Vitamin D is a lipid, so it can cross the plasma membrane very easily, but not so much vitamin C, because vitamin C is water soluble. Okay, so here's an example of simple diffusion and how it depends on its concentration. I think you do an experiment like that in the lab. So you put on this very concentrated blue liquid, and you see that by little by little, it diffuses throughout the water that is in the vase, and then when it reaches equilibrium, then diffusion stops. And you see here how if the concentration is the difference between high concentration and low concentration, if the slope is smaller, then it will take longer than if the slope is greater. So you see how it depends on the concentration differential. Okay, so what about now we move to facilitated diffusion? So facilitated diffusion represents the movement of ions and particles that cannot freely cross the plasma membrane because they can be polar or they can be hydrophobic. They cannot, they're not lipid soluble. So how can these go in and out? They use protein channels. So sodium and potassium, because they're charged, they cannot cross through the phospholipid bilayer. So they're dependent on the channels. Some of these channels are always open, they're not regulated, but others are highly regulated. So for the nerve impulse to go through the axon of neurons, it depends on these highly regulated sodium and potassium channels. But some of these sodium and potassium channels open and close randomly, so they're not regulated. So you see here the protein channels allow charged ions or molecules to go through that are not lipid soluble to cross the plasma membrane. And these are transmembrane proteins. Never forget that these channels are always, they are proteins that provide for this channel. Also, they facilitated diffusion not only relies on proteins, but also on carrier proteins. So I gave you the example of glucose. So here you don't have a channel, you have a transporter. So that whatever wants to cross the plasma membrane will bind to the protein, change its shape, so that then that molecule can go through. Signaling molecules are hormones. There are many types of signaling molecules. There are neurotransmitters, hormones, all kinds. But would the phenylayer be easier to cross? Yes, because you don't have to go such a big distance. So remember, protein channels and carrier proteins, they help molecules that cannot freely cross the plasma membrane to get in and out of the cells. Now, one very, very important mechanism that allows glucose to get into the cell. So you know that glucose is essential. Why? Because glucose is the preferred molecule of fuel that is used to produce ATP through the mitochondria. So on the plasma membrane, we have these proteins, they're transmembrane proteins, and they're glucose transporters. Some of these transporters are not all the time present in the plasma membrane. It's not very good, excuse me. So some of these transporters move in and out of the plasma membrane. Some of the movement of these transporters associated with the plasma membrane, or let's say the association of these transporters with the plasma membrane depends on this hormone known as insulin. Now remember, insulin is a signaling molecule, just like one of you was saying. So insulin is produced by the pancreas. And what does it do? Insulin, its main function is to down regulate, to down the concentration, to lower the concentration of glucose in the blood. And how does it do that? So insulin binds to its receptor. You see, this receptor is a protein that is also transmembrane, but the insulin cannot get into the cell because it's a protein. So the protein cannot cross the plasma membrane. So how does it signal the cell that now it has to increase the number of glucose transporters? How is this possible? So insulin binds to its receptor that is facing the outside of the cell. When glucose binds to its receptor, the receptor changes it. It's a protein, it changes its structure and sends a signal inside the cell. What happens then? This signal induces the transport or the movement of the transporter that now gets embedded on the plasma membrane. So the more insulin, the more the activation of the insulin receptor, the more glucose transporters will be embedded in the plasma membrane. Thus, more glucose will get in from the blood into the cell, and therefore this causes the lowering of glucose in the plasma membrane, in the plasma, or let's say the extracellular space. So you see here a beautiful mechanism where a hormone is released from the pancreas and sends its signal binding. So the hormone is insulin. It binds to its receptor. It originates a signal inside the cell. And what does that signal cause? It causes more glucose transporters to be embedded in the plasma membrane. So if you have more glucose transporters, then you have a better chance of glucose getting into the cell. Now, as soon as the glucose enters the cell, it's phosphorylated by a kinase and it becomes glucose 6-phosphate. So this is an amazing mechanism that was investigated, and it's so important for the health of so many individuals that suffer from diabetes. It's kind of not really positive because the result, well, you start with high glucose levels, so it would be a negative feedback. You start with high glucose levels and you end up lowering high glucose levels, high glucose levels in the blood that stimulate insulin release from the pancreas. Then insulin binds to its receptor, causes more transporters to be embedded on the plasma membrane, and the result is to lower blood glucose. So the stimulus is high glucose, and what happens is to lower glucose levels in the extracellular fluid back to homeostatic levels.
So would this be negative feedback?
It's negative because the trigger is high blood glucose levels, and then it ends up lowering blood glucose levels. So the more insulin, the more glucose is taken out of the blood and into the cell. Exactly. Now remember, this is passive transport. It does not require ATP. So here again, it's a higher magnification of what I just said. Okay, now let's talk about the movement of water. So water also moves in and out of the cell. The movement of water is known as osmosis. So now we're just talking about water. Forget about ions and molecules such as glucose. Take that out of your mind. Just concentrate on osmosis, on water. So Water also diffuses from a region of high concentration to low concentration. Remember, this is ATP independent and it's diffusion. So osmosis is a process of diffusion applicable to water. So let's say that you have these two vases and they're separated from a selectively permeable membrane. It's not semi-permeable, it's selectively permeable. It depends on the charge and the size. Now let's say that this water has particles that cannot cross this selectively permeable membrane. That would be the plasma membrane. Let's say the proteins. Each of these dots, red dots, represents proteins that cannot cross this selectively permeable membrane. So you have water, you have, so water is going to balance this concentration difference. So if you have fewer particles, fewer of these impermeable particles, that means you have more water. If you have more of these impermeable particles, you have less water. So this is critical that you understand that on this side, you have less water particles than in this, because you have more impermeable particles than this side. So remember, for diffusion to occur, so the red particles, they cannot cross, forget it. But the water molecules can cross. So they have to go from a region of high concentration to low concentration. And this is what osmosis is. So that means water is going to move in this direction to balance the concentration on both sides. So this movement of water from high concentration of water to low concentration is osmosis. And this depends on osmotic pressure. So you see here, you have these small spheres represent water particles, and the larger represent the impermeable particles. So you have more impermeable particles here, less impermeable particles here. So you have high osmotic pressure that is going to push water from this side, where there's a lot of water, to this side. So to balance the concentration of water on both sides. So you have to remember here that these large particles cannot cross the plasma membrane, so water has to move to balance the concentration on both sides. And that's what osmosis is, and it depends on osmotic pressure. So I'm going to pass this, and I'm going to move on to this. So osmotic pressure is the movement of water between different body compartments. So here I was using a vase that is separated by a selective permeable membrane. But now let's go here. Why is it that the selective permeable membrane versus a semi-permeable membrane? So semi-permeable or semi-permeable membrane is just dependent on the size of the particles that cross it. A selectively permeable membrane depends on the charge and size. So it's very hard to mimic the selectivity of the plasma membrane. So osmotic pressure regulates the movement of water between different body compartments and also maintains the cell volume. And that's very important. So you know that the blood vessels, the walls, proteins cannot go in and out because they're too big. So the cells that make up the walls of the blood vessels are impermeable to proteins. Remember, during filtration, proteins cannot move in and out. So proteins are stuck either inside or outside. So what happens, water goes in and out to establish a proper concentration and equilibrium. So here you have the interstitial fluid, and here you have the plasma inside, or let's say the blood. Now, let's say that you damage the blood, the walls of the blood vessel. You damage in such a way that proteins can get out. So if proteins can get out, then the concentration of proteins on the outside is higher than inside. So what happens to water? Water moves out, and this is what causes swelling or edema. So when you have a swelling, when let's say you hurt yourself, you hit something, and you get swelling, that means that water is moving in the opposite direction that it should, and it increases the volume in the interstitial fluid. So there's a disequilibrium in protein concentration, so that water moves in the wrong direction and causes the swelling. So you can see how osmotic pressure is so important to maintain the volume, not only of the cells, but also of the organs. So an edema represents a disruption of this osmotic pressure, so that water moves in the opposite direction. Okay, so because of this osmotic pressure and osmosis, we talk about a parameter that is known, or a factor that is known as tonicity. So tonicity represents the concentration of non-permeable particles. So if we go back here, you see the tonicity would represent the concentration of impermeable particles. So I would say that this side has a higher tonicity than this side because it has a higher concentration of impermeable particles. Okay. So remember that tonicity represents the concentration of non-permeable particles. So it's very important that red blood cells, so that the blood concentration of impermeable particles is the same as the fluid inside the red blood cells, so that there is no change in shape. So if the red blood cells would be in a hypotonic solution, a solution that has a lower concentration of impermeable particles, then water would come out. I mean, water would go into the red blood cells, and the red blood cells would burst through a mechanism known as hemolysis. So remember that water always goes in the direction of high concentration to low concentration. So if the solution that is outside the red blood cells has a lower concentration than inside, then water will go in I'm so sorry. And will cause the cells, the red blood cells, to burst. What about the opposite? If the outside in the blood, the concentration of impermeable particles is higher than inside, then the water will move out, and this will cause crenation. This red blood cells would shrink. So why am I telling you this? Because when you get a transfusion, a blood transfusion, you always want, or when you put an IV in the patient, you always want that solution, for example, to deliver some medicine, you always want that solution that's going in the IV to be isotonic. That means that the concentration of impermeable particles is the same as the fluid in the red blood cells. Why is that important? So that you don't get the bursting, you don't get hemolysis, or you don't get crenation, because the tonicity of that solution that is delivered through the IV, the concentration of impermeable particles is the same as in the red blood cells. So the IV Most intravenous solutions are isotonic. What do they contain? Either 0.9% of sodium or 5% of glucose. So this equilibrates, is the same concentration of impermeable particles than inside the red blood cells. And therefore, the shape of the red blood cells will not change. There's no attendance for the lecture. Okay, so what happens in dialysis? So dialysis is a way of purifying the blood independently of the kidney. So if the kidney is not working properly, then it can lead to changes in pH. Waste products will accumulate in the blood because they're not being filtered through the kidney. There's a concentration of nitrogenous waste. Nitrogen results from the breakdown of proteins and nucleic acids, and they have to be removed. If they're not removed by the kidney, this leads to azotemia. So dialysis is a way of artificially clearing the blood independently of the kidney. So you have the blood go through the tube that brings the blood into a bag that is, in this case, selectively semi-permeable. And it's placed in a bath that contains ingredients that make it isotonic. So this way, the blood is going to be, the blood in this bag that is being pumped because of the pumping of the heart is going to be cleared in this semi-permeable bag. So you have glucose and buffers that come in that are beneficial, and you get rid of the nitrogenous waste in creatinine and urea. So it's a way of purifying the blood independently of the kidney. So we talked about active and diffusion through the plasma membrane, so passive transport. So we had simple diffusion, carrier through carrier molecules, so facilitated diffusion. So through carrier and channels, then osmosis. Okay, now I want to move to active transport. So there are two types of active transport. Remember, in active transport, the particles that are being transported move from high concentration to low concentration. And this movement against the concentration gradient requires potential energy in the form of ATP. So active transport per se involves smaller molecules, smaller substances. When they are big substances, then this is vesicular transport. And we're going to cover this today, the active transport. So there are two types of active transport, primary and secondary. In the primary active transport, that means that the protein is responsible oh my gosh. Yes. So active transport goes from low to high, right? In the active transport, the transport goes from low to high against its concentration gradient. And that's why it requires ATP. So the secondary active transport, that means that the transporter relies on the primary active transport. So the secondary transport does not require ATP, but it's an active transport because it's dependent on this pump. So let's see. In the cell, in relation to potassium and sodium, the inside of the cell contains different concentrations of these two ions. So inside, the concentration of potassium is very, very high, 139 millimolars. The outside, in the interstitial fluid, the concentration is 4 millimolar. And sodium is the opposite. It's high concentration on the outside and low concentration in the inside. So this is maintained by active transport. That means the potassium is moving inside and sodium is moving outside to maintain this highly differential concentration. So this is dependent on what we call the sodium-potassium pump. Now, active transport, so you have two molecules that are moving in different directions. If it moves just one molecule, that means it's a uniport. If there's two molecules, that means that are moving in the same direction, that means it's a symport. So here you see two ions that are moving in the same direction. An antiport, the molecules are moving in the opposite direction. So I just want to cover this and then we'll stop. This is a very, very important protein that is present on the plasma membrane. It uses up 40% of the ATP that our cells produce. And what does it do? This is a sodium-potassium pump that maintains this differential concentration inside and outside of the cell. So you have high concentration of sodium on the outside and high concentration of potassium inside. And this is possible because of this pump. So this pump moves 3 sodium out and 2 potassium in. So you have high concentration. I cannot do this anymore. Sorry. This is ridiculous. Oh, sorry. Okay, so just remember about this sodium-potassium pump. It's very, very important to maintain the sodium and potassium concentration on opposite sides of the plasma membrane. Okay, I think we'll stop here. Okay, so any questions about the exam soft? We'll stop here. I think, yes,
Professor?
let me just get a little bit of water. Ridiculous. Okay, so questions?
Professor, can you
Yes.
show us how we can sign up or log in on the exam soft using your screen?
Okay, so I'm not going to do this because you should try it at home. And if you have problems, you can ask the support group of ExamSoft. So there's no use for me to do this here because it would take a long time. And I think you can do it and try it at home and contact the support group. Okay?
Oh, because when I tried logging in, it told me wrong password, and I'm trying to reset the password, but it's not working.
Okay, so the best thing, I cannot do anything about that. Contact the technical service. As I said, they are excellent. So you can call them on the phone or chat with them. I think that's the best thing, the best suggestion I can give you.
Professor, so you're saying that we have to download the software?
Yes.
Professor, my name is Nayeon Kim. Today's first day. So what is the sphere of for our exam one? You told me that on Thursday we have an exam, right?
Well, it's a mock exam. You have to come, everybody has to come to Hunter, to the assembly hall,
Uh-huh. And how
so
can
that you
I
can
get
try out. But you should,
it's not exam one, just mock exam.
it's mock exam, not exam one.
All right. And how can I get the PPT? What you teach this one?
The what you mean the PPT?
I mean PowerPoint. What you show? What you
Yes,
show
it's already
us?
downloaded.
A download? Really?
Yes, it's already
First
posted
space?
on our
Oh,
right space.
yeah. Okay, I will find it. Thank you. And our sphere of exam one or mock exam is chapter 1 to until when? But until what chapter?
Everything is listed on the syllabus. It's very, very
Syllabus.
detailed. So look at the
All right.
syllabus, okay?
Okay, got it. Thank you.
Professor, you said we could do the exam now if we're able to log in?
Yes, but the exam is a mock exam. It
I
has,
know. I just wanted to be sure if we can
yes,
just.
you can try it whenever
Okay.
you want, and you can try it more than once
Okay.
and try on your own. And then I sent everybody the instruction in an email. I sent you the instructions in
All
a
right.
very detailed manner.
Okay. Be well. Just get
Professor,
some hot tea, Lipton and some ginger. You'll be fine. Bye
Okay.
now. Bye, Professor.
Okay, goodbye.
Professor, if we take the mock exam online, then we don't have to come on Thursday.
You have to come on, you have to come. I want to see everybody there because there's going to be 700 students. And I want to make sure that everything works when you are in the
Professor-
auditorium.
Professor, I have one question more. Do we have to pay $180 for person?
Well, that's your decision. That's not my decision.
I don't need to use Pearson, just
It's up to
I
you.
don't need to. All right. And also
your-
ebook. For ebook, do I have to pay?
You have to have mastering. It helps a lot.
Uh-huh.
And then some exercises.
So I need to pay an ebook and read ebook. Do we have to? Or if I study for your
As I said, this is your decision.
right. So
I'm not going
on
to tell you
Thursday,
what to do.
Alright. On Thursday, if we have a mock exam and then we can know how we study or prepare of exam one, right?
So on Thursday, half of the
Mhm.
class,
Mhm.
I should say half
Okay.
of the time, but I'm also going to cover some material on Thursday.
The material.
And this won't be, and this will be in the auditorium.
Oh yeah, auditorium. We have a mock exam. We can have a test. We
Yes,
can know.
but
Okay.
I'm
Mhm.
also going to cover some material. Okay,
All right. All right. On Thursday. See you on Thursday. Thank you.
so everybody should be present on Thursday in the auditorium in person.
All right.
Okay,
Professor, you said the exam soft is
yes.
going to be on Brightspace. Where can I find it in Brightspace?
Can you ask that question again? Sorry.
You said that the exam soft is going to be in Brightspace. Where
No.
can I see it?
Exam soft.
Yeah.
So read the instructions. I send you instructions on how to access the exam. I send an email. So you have to go to your Hunter email. And you have to sign up to download the ExamSoft program. And then you will have access to the mock exam.
Okay, thank you. Feel better.
Okay, thank you.
Professor,
Thank you
just a
so
quick
much.
question. Do we need to have a device that records your video? Like, are you guys gonna record our video during the
Sorry?
exam? During the exam, will our video be recorded? I just need to know so I bring in a device that has a working camera.
Which I don't understand what.
So
what is being-
some Proctor softwares they record your video. So will ExamSoft record our video? Like, should I
No.
have a device that? Okay.
But
Okay.
there will be proctors in the room.
Yeah. Okay. Thank you.
No, it won't record you. No,
Just making sure so I have a working camera. But thank you. Feel better.
thank you.
Professor, and it will be the same time, right? 4 to 5:15.
Yes.
Okay. All right, cool.
Make sure that your devices are charged.
Okay.
Don't come with your devices
Dead.
with
Okay.
almost no charge. So
All right. Thank you.
Professor, I tried contacting the tech team, but no one is responding, and I don't know why I'm not able to like.
you tried calling or the chat?
I tried calling and no one's responding, and some people in the chat box wrote that the tech team does not help like properly.
Maybe everybody called at the same time. I will
Like-
send them a message.
Has anyone like successfully logged in? Like, how did you do that?
Yes.
Yes, I went to my Hunter email. I clicked on the link. I put my school email,
A
and
lot
then
of people have signed in. So you have to activate your account. I send them an email with all the instructions. They're also posted on the website. There's an announcement on the Brightspace website that has the instructions, okay? Step by step.
yeah, but when I'm clicking on the link, it's not taking me to. Okay, hold on. Let me private.
Well, I'm not going to wait here for you to try, sorry. You have to try it on your own, and then if you have a problem, contact their service.
I have a question now.
Yes,
Professor. So I logged in successfully and everything. I see two courses. It says anatomy and physiology 1 and exam soft training for students. You said that we're able to do a mock exam now. I click into both of the courses. I do see a mock exam, but I can't access it.
you have to use the password.
Yeah, there's nothing for me to even put the password in. I click into the courses.
Devin,
I
please,
yep.
please contact the tech support. They will help you.
All right, then, and what's this Exemplify thing? Do I need to download this Exemplify?
Yes, you have to have exemplify. Yes.
Oh, okay, all right. Thank you then. Thank you. That's it. Have a nice day.
Okay.
Hello, Professor. I had a quick question. Am I allowed to use an iPad for the mock exam?
Yes.
Okay, thank you.
No iPhone. You can use an iPad.
Okay.
Professor, I had a question. So I'm currently having issues for a couple of weeks, though, since kind of school started with my CUNY login and my email itself. So I don't know, something happened to it. I was able to go to the registered office today and get it kind of fixed, but I haven't they told me to wait till tomorrow to try again and see if I'm able to log in. So I haven't been able to really see any of like the announcements you've mentioned about the mock exam and everything. And I
The
was able to
mock
get into
exam,
class
the
because of a
instructions,
couple friends.
Madeline, for the instructions for the mock exam, I sent to your email.
Yeah, exactly. My email is also not working. Like my CUNY login and my Hunter email itself, I don't know what happened to it, but it's not working. So I'm not receiving anything from my professors. The way I find out is by like my friends from the classes. And I can't do the exam soft thing because I need to do it with my CUNY login from what I'm seeing from the other students are saying as well.
Yes,
So
but you said it
hopefully
will
by
be,
tomorrow it does
yes,
work, but
yes.
if anything, I'm just trying to let you know and give you a heads up about it.
Well, that you don't have to let me know because this is yours, your problem, right? You have to fix it. Hopefully by tomorrow it will be fixed. If not, contact the registrar again, okay?
All right. Thank
Hopefully
you.
everything will be working out.
Mm-hmm.
Okay? Okay, anything else? I apologize for my cough. Okay, bye-bye.